Pollard's sneaking in the back door. Okay, so that's Miss Pollard. She's our lab prep person. She's the one you have to thank for all the media we have to grow our bacteria in. Yeah. She's also the shoe Nazi, though. Did she run away already? Okay. So she's our safety officer, so as well. So lab 1, we're going to talk about some materials that are found on page 6 in your lab manual, which is the value of controls, right, in experiments. And then we're going to go over what we just did, which was ubiquity of microorganisms. And then we're going to introduce the light microscope, which you should already be familiar with, right? We're going to go into maybe a little bit more detail than you have in your previous classes as it regards to the microscope. Because as you can imagine, it's a very integral part of this course. And you will actually have your very own microscope that you will use. So, false positive. What does that mean? That means that you're dealing with probably a control, right, that you know should come out positive, but in fact it came out negative, right? So where the observed positive result is not there, instead you're getting a negative result. So probably the easiest test that most people are aware of to help you understand this concept of false positive and false negative results is pregnancy tests. So, false negative, as it implies, right, is where the result is negative when in fact you expected a positive result. So, if you're pregnant and you run a pregnancy test, you expect it to come out positive, right? What if it comes out negative? Does that mean you're not pregnant? No. It's dealing with one of the things about the tests, and that is sensitivity. Usually when you have a false negative result, when you're expecting a positive, it's an issue of sensitivity, the limit of detectability. How good is that test at detecting what you're looking for? What are its limitations? So in the case of the pregnancy test, and also if you can't listen to me and write at the same time, which I don't know anyone who can truly do that, this PowerPoint will be posted for you guys, and this recording. So don't feel the need you have to write, because I'm going to go fast too. Okay? So, do what you feel comfortable with. It'll help you retain information. So, for sensitivity. So, when you run a pregnancy test, you're testing a urine sample for the presence of human chorionic growth hormone, which is produced when a woman is pregnant. It's produced at higher and higher levels throughout pregnancy. So, one of the things that they tell you is that you have to wait a certain time period before you can run this test for it to be accurate, right? And that's when normally your levels will be high enough that it's within the limits of this test, okay, in your urine. What would be a more accurate test that you could test earlier, where the hormone level would be higher? Blood test. That's why blood tests can tell you sooner, right? Because that hormone's in your blood. It's only when it's filtered out of your blood, right, and that it ends up into your urine and leaving your body. Make sense? So that's the limit of sensitivity, right? You have to have enough of the hormone to be able to get that positive result that you expect when you're pregnant. Make sense? Okay. So again, that's a sensitivity issue where sometimes you'll get false negatives. Where in fact you are what that test should be testing for. You're in fact pregnant. Now the flip side, right? If it comes out positive, don't take 3 or 4 or 6 more tests like Juno did, right? You're pregnant, right? It's a pretty specific test as far as women are concerned, right? If it's coming out positive, right, you're producing this hormone, probably because you're pregnant or some other issue, right? But it's pretty specific for this hormone as it relates to women, Make sense? So there's no sensitivity issue there for women. Men, on the other hand, can they get pregnant? No. Currently, no. Can they get a positive result in the pregnancy test? They can. They can't. Because this test detects something else that will sometimes be present for men. How or why they figured this out, I don't know if a guy felt bad for the woman, was like, I'll pee in the cup too, or what, I don't know. But they found that there is a particular substance, I don't know exactly what, I haven't done further research on it, but it is a true story, I'm not lying to you, that men, when they have, I always struggle on this word, testicular cancer, produce a substance that can be detected by the pregnancy test. So it can be a pre-screen for this type of cancer. I'm not even going to try and say it. I'm twisted on that one. Okay, so we all know about the big bad C word, right? Good old cancer. So for women, right, we do breast exams every month, right, ladies? Trust me, do it. My 35-year-old sister found her own lump, lost her breasts because of it, but didn't have to go through chemo or radiation because she caught it early, right? So don't skip it. Mark it on the calendar, right? Get it done in the shower, wherever you need to do it, right? But get it done. And then when you get old enough, or depending on your situation, right, go for the boob squeeze once a year. Okay. And the guys are laughing, right? They're like, ha, 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 right, right. Actually, guys can get breast cancer too. Guys should do their own exams. Of course, depending on how much body fat and whatnot men have, right, it can be more difficult, like it is for us, to do such things or detect such things. But you don't have to stop there either. You should also do your testicular exam every month as well, right? I've had a friend who's a professor here, right, lost one of his testicles to good old cancer, right? So, and then women, we can't check one other thing that we need to check on a yearly basis. What's that one? Good old cervix, right? Somebody else has to do that for you. I don't know about you, I just don't look at her in the mirror. Just don't, right? So you got to go for that check every year, right? Because you don't want to deal with that either. Okay? So keep up, right? Take care of yourselves, right? So you can keep coming back to class, right? And we can have a good old time. Make sense for everybody? Okay. So, controls and standards. Standards are a little bit more involved controls. What we did today, and what we do most of the time in this lab, is just run standard controls where we're doing a qualitative type test where we're getting yes or no results, right? Positive or negative. But standards are used a lot in laboratory settings, even some of the tests in which you may have run, right, on your blood or different products from your body. And these require standard because we want to know quantity, right? When we're doing quantitative testing, we want a number assigned. We don't want to know whether you're producing human chorionic growth hormone. We want to know how much you've produced, right? What level are you at, right? And for that, our control needs to help us determine that. So our controls have to have all the information. So we have to have multiple controls of different concentrations to help us narrow down the unknown sample's concentration, right? So when we don't know what a sample is, right, and help us get to that value. So this is an example of a protein assay. And again, in this class, we're not going to go to this level currently, but we are working on getting the technology and the equipment where when one of the tests that we run, we could actually quantify it instead of a yes or no. So this is an example of a test well plate where there's 96 tiny little wells here, so almost like 96 little test tubes. And that scientists can run tests in. And a lot of the tests that we run will produce a color change, or you'll have turbidity that can be measured by a special spectrophotometer. We stick the whole plate in, right? And the machine looks at each well and is going to assign a number value to each one of those wells based on the color or how cloudy that well is. And so what you have here is a reading from a machine like a spectrophotometer on the absorbance rate at a particular filter color. And then their known amounts of sample. So 4 micrograms of the sample. Can somebody just close that plate for me? And then what the reading came off for that machine. Okay, so we have a set of standards, right? A set of controls to create our standard. So we have 4 milligrams, 8, 12, 16, 20. So when I would run tests when I worked in a research lab, I was looking for antibody content for the armadillos that we infected with leprosy. And so I would take a blood sample, and the antibodies in that sample, I would quantify about how many antibodies I got from each sample to know where they were in the progression of the disease. And we're going to run a similar test to that, where you can detect the presence of antibodies in the last lab of this semester. But again, we're just going to be asking yes or no. We are not going to put it on the spectrophotometer and actually get a number. But we could do that, and we're trying to work towards that. So you guys get to do stuff that real scientists do. Right? Not to say that we don't do yes or no, right? The same technology is used in pregnancy tests, and again, we really just only care, are you or are you not, right? So sometimes yes or no is enough. Other times we need quantitative values, right? And so we would plug these numbers into a computer, right, into a program, and it would help calculate, right? We could do a linear regression, which, as you notice, they're not all lining up, right? Or we can do a little bit more complicated math where we can take into account all the points, right, and make the line match up to all the points. And of course, depending on which you do, your values are going to vary differently, right? Because for an unknown sample, if it comes in here, one of the lines, it hits it at a particular concentration, and at the next line, it hits at a different concentration. Right? So it just depends on how specific the scientist needs, how accurate do the values need to be as to what type of mathematical equations they'll use. One of the other professors that teaches this lab likes to go into all the math. I like to show you guys, some of us are mathematically minded like him, right? But this isn't a math class, right? So although we are going to do some math today, but it's not going to be too high tech, right? And we're going to do some more later that's kind of difficult, so we'll really work through it together. But he has all the math there for those of you guys that, like, you see this and it makes sense to you, right? If it doesn't, don't panic. Okay, so in your lab manual, they have like little pictorial representations. So I've defined these things. I've given you an example. I've shown it to you mathematically, some of this information, right? So we're going to present it in all the different ways that hopefully learning style-wise we're going to get it across to you. So if your observed result is negative, right, and you thought it was supposed to be positive for your control, this is not good news, right? And this is probably, again, as we said, a sensitivity issue, right? If you're getting a negative result, it's probably not a negative sample. We just aren't getting within the limitations of that test, right? You probably don't have enough of that substance to tip it over so that this test says you're positive. Make sense? Okay. So, frowny face for that. If it's supposed to be positive, it comes out positive, great, right? And there is a positive control built into every pregnancy test. Even the ones with the cute little digital readouts, if you take it off, you'll have 2 lines if you're pregnant. One of them was your unknown sample, which, oh, you're pregnant, it came out positive. And the other is the positive control for that test to make sure the test is working properly. If you don't get that test well line, it says error, repeat test, right? Because it can't tell you whether you're pregnant or not because it couldn't even figure out if its positive control was positive. Make sense? Okay, so we run controls, right, when we run these tests, just as today, right? We want to make sure that media we started with was sterile. So we kept it closed, and nothing should grow on it, right? Because that means we started with sterile, non-contaminated food. Okay, if we have a observed positive result and it was supposed to be negative, It's specificity issues. Something else, right, other than what we thought we were testing for is causing it to be positive. Again, this is bad news. So this is the case of the guy, right? Guys aren't supposed to be pregnant. How the heck did his test come out positive? Well, he's not producing that particular hormone, but a substance close enough that the test is thinking, right, that it's there. It's positive. We could mathematically calculate these, of course, right? And this is where you have, unfortunately, an inaccuracy in your lab manual as far as specificity goes. Remember, the issue with sensitivity is false negatives, right? So the less and less false negatives we have, the more likely we're going to have nothing but true positives. We're going to have a 1-to-1 ratio, right? And we're going to have a very sensitive test, right? Which is the case as far as pregnancy goes for women. That's a really pretty sensitive test. And they're improving all the time. Specificity, right, is how specific that test is testing for exactly what you're looking for. And so this is where sometimes, unfortunately, you're getting false positives. You don't want those. The less and less false positives you have, the more true negative results you have, the more likely you have a very specific test. And again, specificity with the pregnancy test and males, right? And that it's something else we're actually detecting, right? You're getting that positive result. So we have some terminology as far as ubiquity of microorganisms. What does ubiquity mean? That's a weird term, huh? It means that it can be found just about anywhere. Ubiquitous means everywhere. And this is referring to microorganisms in general, right? You could find them just about anywhere on this planet, on our bodies, within our bodies, you name it. It's in the air, right? Microorganisms are in the air we're breathing right now. Which is why you should breathe mostly through your nose. It's your filter. Keep most of that bad stuff from getting all the way in. Pathogens. What do we think when I say pathogen? Disease, illness, right? And this is an organism that we know causes disease, right? We know will make you sick. Klebsiella pneumoniae. We know it will cause a serious form of pneumonia. Do not lick the petri dish. Non-pathogenic. Well, non means not, right? So these are ones, though, caveat in science, though, organisms that are not known to cause disease. Right? So right now we might say something is non-pathogenic. Does that mean that it definitely is not going to cause disease? No, it's just that we haven't found that it causes any disease at this point. Later on we may discover, oh, it causes this disease. We didn't know. Right? Science is ever changing. So is history, by the way. My ex-husband was a history major in grad school. History is really interpretive. It's, you know, like we think, you know, it is what it is. Well, it's based on what people said happened. Right? Different people say different things, and they discover, right, in the past, I was like, oh, it didn't really happen like that, did it? I was like, man. And just when you think you have something definitive. That's why I love math. Right? Math, you get it, this answer 6 different ways, but there's always a right answer, right? In science, not so much, right? Don't get fixated on that right answer in science. You'll be disappointed someday, right? Because we're going to discover something else and find out, oh, well, yeah, this is true. And that wasn't true. Right? So, opportunistic pathogen. So again, we're thinking disease because we're saying pathogen, right? But not always, right? When the opportunity arises. So some of these guys are only bad guys when there's the opportunity to be a bad guy. You see? Who tends to be bad guys under those circumstances? Good old enteric bacteria. So who's enteric? This is an informal name given to microorganisms that live in the gut, the intestines, right? Of warm-blooded mammals like ourselves. For instance, E. coli. Your large intestine is chock full of it right now. Per gram of feces, you have about 1 billion of bacteria. The good news is in your intestines, in your large intestines, it does not cause you any problems. In fact, they even benefit you. The problem is when poop gets elsewhere, when these bacteria get in different parts of your body. So little girls have this problem. We have 3 holes called a urethra, a vagina, and an anus. Poop comes out the anus, and when we wipe it away, we have to wipe it back, because if you go front, you could wipe it into your vagina or your urethra, and therefore contaminate those 2 environments with microorganisms from your intestines. These can cause serious life-threatening infections. A little girl in Louisiana several years back died of something as simple as that, not wiping properly. Bacteria got introduced into her bladder, traveled up her ureters to her kidneys, and killed her. Her parents did not know what was wrong with her, did not seek medical help soon enough, and she died from something that simple. So guys, don't tune me out, because someday you may be a single dad to a little beautiful girl. You have to teach her these things if you want to keep her. Guys don't have that problem, right? 2 holes. Distance varies, but there's still quite a bit about a distance there. Not an issue for you guys. Alright, I'm going to slightly not gross you guys out. I'm trying to keep it as PC as I can. You'll have to fill in the blanks. A student of mine in microbiology class was babysitting his son. Wasn't doing a very good job paying attention to him. Came back into the room and poop is everywhere, including having been ingested. He's like, how come he didn't get sick? I'm like, the good news is your poo going right back into your poop tract is not a big deal. It's getting into other places. Right? That was his poop that he ate. It went right back into his digestive tract. No problem. Stick it up your nose, stick it in other places, right, where it's not supposed to go, you could create infections. Right? Rub it into a wound, not a good idea. But your own bacteria are good news for you if they go where they're supposed to go. Alright. Like I said, I tried to keep it, but it's real stuff. Alright, really happens. Okay. In animals, when they ingest their own poo, it's usually because they're not getting the correct diet that they need. All right, you need to change their diet. But animals will eat their parents, like their mom's poop, and again, that's to establish the microbes in their digestive tract. That's a normal thing. Cows and horses, you don't want to stop babies from doing that. And they naturally pick up the microbes from the environment from sucking on their mom's teats and stuff like that, and establish the microbial environment they need. So that's also why being too clean can be bad, right? Because you actually acquire microbes in your environment that help you. So if you were in a sterile environment, you'd die. Right. Unless you had to be in a sterile environment because your immune system isn't working. That would be the bubble boy. Right? Non-functioning immune system. Okay, so host, right? If you're a host, right, you have visitors, right? Things that have come over. In this case, you are the habitat, if you're the host, for another organism. Usually when we're talking about hosts, we're talking about parasitic type relationships or commusal relationships, which I believe there really isn't a such thing as commusal, but I don't know. They say there is. Commusal means that it's a synergistic relationship, so you're living in the same place, right? Your host, and the relationship is such that the other each other are not affected negatively nor positively. You're living harmoniously, right? There's no negative side effects of living together. There's no positive. I don't believe that's possible. Parasitic, we know, is bad news, right? It means that the host is negatively affected by the organism. Okay? So my son was an endoparasite, now he's an ectoparasite. I'm just kidding. My parasitologist friend, this is how she refers to my family. Right? But we do more for our kids than they do for us sometimes, right? But he gives the best hugs, and it makes up for all that I do for him. All the money he cost me. My husband was furious last night. He broke his shoes. He was bored and he broke his shoes. I'm like, dude. School shoes nowadays, they have to be black or white. That's not an easy task. Right? I was bored. We go mow the lawn. We don't have a lawn. I live in the city. Okay, mutualistic on the other hand, and I always think of marriage, right? At least a good marriage. In this case, both partners should benefit, yes. Right? Both should benefit. And as I said, the microorganisms in your gut, you're feeding them, you're giving them a place to live, and they're producing vitamins for you. They're helping digest some of your food you don't digest. That's a mutualistic relationship. They say sometimes it's a communal relationship with the organisms that live on our skin. But I really think it's more of a mutualistic because, again, they have a place to live, they have food to eat, and they're helping you stay disease-free by being there. By them taking up the space and eating the food that's available on your skin, pathogens don't have any place to land and nothing to eat. So that's really a benefit to you. A reservoir. So this is a non-human host, right? So some other animal other than a human, right? We forget sometimes we're animals, right? We put ourselves on this little pedestal. So non-humans, right? So other animals, or other sites in nature that serve as a perpetual source of pathogenic organisms, right? What do you want to avoid? In Louisiana, as far as rabies is concerned, anyone know who you should avoid? Who's the #1 carrier? Bats are bad, but even worse. Raccoons. Yep, raccoons. Leave those cute little guys alone. Right, so we know these things, right, and we can avoid them or protect ourselves, potentially. Free living, right, they don't require a host, a specific host, right? So these could be found in the environment. They could be on or within a person, but they don't necessarily have to be there. And therefore, they tend not to be pathogenic, right? Because if you require a specific host, right, then you've got to be there. You can't live on your own. So what was our specific purpose of incubating our unopened plates? These are our what? These are our controls to show that the media is sterile to start. So remember, what's in these plates? If we don't open it, it's just the food, right? Just the media. That's it. So our label for these would be negative control or no exposure. What other important information do we need to include on a lot of the things that we do in this lab? What did we add to, what other information did we write on these plates? The date. What else? The temperature, right? Because we have different incubators in this lab. One more. Are we the only class? Are we the only people using this lab? No. So if it's your plate, you need your name on it. If it's the class's plate, like today, right, we need some identifier that we know it's our class, right? I'm horrible with section numbers, so unlike in the past, I just wrote Wednesday because we're the only Wednesday lab. Or had you guys write Wednesday. Thank you. And as you can see, we write along the edge and the bottom of the plate so we can clearly see what's on the plate. It's okay if you didn't write on the edge, right? But from now on, it's good practice to write along the edge of the plate. Helps in the visualization of what's on the plate. So, what does it mean? Right? So if growth appears on both our unopened plates, what happened? What went wrong? The control was contaminated, right? Our media was contaminated. Either the person who made it, right, or the person who poured it into those plates, right, may have done it improperly or may have become contaminated. The reason why I was so excited earlier that there was no growth on my control plates is that set of plates that I used for my Tuesday lab was potentially contaminated. There was a tiny hole, and the top of the flask when Mr. Brent went to go pour the plates last week, and so we made a note of that, as good scientists would. And I said, well, I have 2 classes, so I'm going to do a little side experiment. I'm going to do possible contaminated plates for one class, and ones that we know are good for my other class. And the good news is that they didn't grow anything, right? So we didn't get any contamination. So we know that when we look at those plates that say fingers, that those bacteria came from the fingers, not from some type of contamination of our media, right? Which is important. So if you're working in a research or a clinical lab and we got growth on those plates, what would we have to do? We'd have to redo it, right? It would invalidate our results, would it not? Right? We wouldn't know if that stuff came from the fingers or from the contamination of the media. We just wouldn't know. So what if only growth appears on one plate? Where would that have happened and why? So it's probably not the whole batch of media that's bad, right? Could have been the preparation of that plate. Or remember what did I do when I was setting up those plates? What did I do for my controls that I didn't do for the other plates? I taped it shut right away so it didn't accidentally get opened, right? So we took special precautions, right? So one of the things that could have gone wrong is maybe one of them could have gotten opened before I taped it shut or something like that, right? Or when the person was pouring the media, right? So it was, or even during incubation, right? But again, one of the ways I avoided that was by taping it shut. Any growth, remember, creates a problem for us on our controls because they're supposed to be negative, supposed to be no growth. Any growth on either one of those plates nullifies our experiment because we don't know, right, if on our exposed plates it came from the exposure or the contamination. The good news is this is a teaching lab, so if we get growth on our control plates, we're not repeating this experiment. This experiment is designed to, and as you guys saw, right, some of the growth that we saw on yesterday's plates, and these are some pictures from past semesters. And this was a growth that I was hoping to find, but didn't have any. And partly because we have a HEPA filter running in this lab to help with contamination from this type of organism. And anyone know what type of organism has that fuzzy type appearance when it grows on the agar plates? Fungus, mold, right? Yeah, mold spores. So mold spores would land on it, right, and then grow. And they produce these aerial hyphae. We'll look at them under the microscope next time, and they release spores. So also, if we get any of those, I usually tape those plates shut. We don't want to open them up and release those spores. Anybody has allergies like myself or sensitivities to mold spores. We don't want to expose people to that. So they asked why. And if you read your lab manual, you'll notice that I actually changed it up a little bit. We did a little bit different exposures than what they have there. The coughing one never comes out good. That's why I've abandoned it. And the hair scratching one too. And nobody really wants to do those two, and I don't blame them. And it's still kind of hard to get you guys to do the mouth and nose sometimes. But those always come out good because those are places where you have microorganisms naturally. So like I said, your nose is your filter. It's continually filtering the ones in the air. And then, of course, you have ones associated with your mouth. So we wanted to demonstrate to you, right, that these organisms are present even though you don't see them, right? And sources of contamination when we start working with our cultures and we want to keep them from being contaminated. We know our fingers are a source of contamination. Our desktop could be a source of contamination, right? Anything we track in from outside, right, could be a source of contamination. The air even could be a source of contamination. So the way we work with our cultures, most of you guys that inoculated plates today, I had you hold the lid above the plate so that you would minimize the amount of microorganisms falling out of the air onto that plate and contaminating our cultures. The reason why you can see so many of them here, right, is that we give them a lot of food, right, and put them under ideal conditions like a nice warm incubator so they grow to very large numbers. You're looking at millions upon billions of bacteria and fungi on these plates. So temperature matters, right? We know this, right? 25 versus 37 degrees. The two main temperatures we use in this lab, other than this other incubator, which is 30 degrees. So why are we doing two different temperatures? Right? Organisms are affected by temperature. And we also want to simulate the best temperature possible based on our samples. So Celsius is what 35 and 37 and 30 incubators in this lab are set at, the Celsius scale. This is based on the properties of water. Water boils at the 100 degrees Celsius or 212 degrees Fahrenheit. And it freezes at 0 degrees Celsius or 32 degrees Fahrenheit. Our 37 degree incubator is the one we use a lot. We use a lot of organisms that would normally grow in our bodies, which is again is why we want to be cautious in this lab. And some of them, in fact, can be quite pathogenic if you were to lick the petri dish, which you're not going to do. Right? Not to mention we don't even use petri dishes that much for stock cultures. They take up too much space and the lids come off easily, right? So we actually use tubes with solid cultures in them, what we call slants. So 37 gets used a lot. It'll get nice and smelly and stinky. And again, the reason why it's set at 37 degrees Celsius is equivalent to around 98.6 degrees core body temperature, right? Nice and toasty warm. Keep our bugs nice and happy. 30 degrees, which is around 86 degrees Fahrenheit, is equivalent to soil temperature or our skin temperature, right? As you move away from your core body cools. Your skin is actually a way of cooling your body. Your blood runs to your skin when you're highly active. That's why you become red, because of increased blood flow. And this is to get rid of the heat that you're producing. So African and Asian elephants have different sized ears, right? Anyone know what we have at our zoo? We have the Asian. They get the little ears, right? African elephants, really warm climate, right? Very large ears, large surface area, so that they can cool their bodies down, right? So they pump a lot of their blood through their ears, and that's why they're fanning them, right? To create convection and cool themselves. Okay? So we're much cooler at the surface. So we work with a few microorganisms that don't like it quite so warm as 37, but need some warmth to grow effectively. And mostly they're soil, what would be soil contaminants that we utilize. But it would also be similar to skin temperature, something that would grow on our skin. Room temperature, which I'm happy to say that is staying around 77. There are times that this room used to peak around 80, and that incubator was never, that one, the heating element usually never turns on. But we don't have refrigeration with the incubators, right? So it was never cooling it back down. So even though we had 35 written on there, it never would read on the digital readout. But right now it is. So you know it's around 77 degrees in here, which is actually nice compared to how it was. Of course, most of us would prefer cooler, right? 75 is my favorite number, but not so in this room or in this building, in this location. So what's the likely source of 37 degrees? It's probably something from us, right? Some organism that came from us. Can it grow at room temperatures? Yeah. Does it have a lot of nutrients at room temperature? Not really. There are nutrients possibly available that you're not aware of, right? Things that fall out of the air, things that are around that we track in. All right, but most of them that are contaminating are coming from us. And the most common offender is actually our breath. And so I actually have a really cool plate that I'll show you guys later on in the semester, and it has a contaminant on it. And it probably came from somebody breathing on their plate. So our desktops, right? You're definitely going to see organisms. And again, because we're going to put them nice and warm, we're going to give them lots of food, right? So they're there, but again, there are nutrients there, but they're not growing to huge enough numbers that you can see them. without the aid of microscopes. Also, microorganisms are really good at starving, or going into states of stasis where they're not metabolically active and don't need a lot of nutrients. Some of them are really good specialists at this to an even greater degree. Some bacteria can form endospores, which are completely a dormant form of that cell. It's protected its DNA. It's waiting for things to become better. Those of you guys that are in my lecture class, we talked about an organism that can do that, can be highly pathogenic. Remember what disease it causes? It's the first one on our list. Anthrax. Anthrax, right? That powder is endospores, right? They can be ingested by someone or inhaled by someone, and that will germinate and cause that particular disease. Others can form spores, right, which can be dispersed, and that's usually a form of reproduction, but also it can survive, right, until food becomes available. Cysts, some of them can encyst, right, and be protected in the environment. They're not growing, right, they're resting, but they're highly resistant to environmental conditions, right, so they're not killed while they're waiting for it to be picked up by the next host. So this is the experiment we did today, right, and I'll take them out of the incubator tomorrow or the next day so they don't overgrow, put them in the refrigerator, and we'll see the different types of growths that we get from the different exposures we did and compare the different temperatures. Having said that, these are some pictures from past semesters, and you can see that, again, we're doing the desktop at two different temperatures, 25 and 37, and different organisms are going to grow best at different temperatures. And of course, the ones that come from us are going to be happy at what temperature? 37. So you'll notice that this 37 plate looks very familiar to the fingers plate, even though we put it at 25, right? It was because it was one everybody touched. We still got a lot of growth. Look at the air contamination in the desktop, right? These look very similar. Look at, they have both these pink colonies and these yellowish colonies, right? Similar type organisms. Again, probably the contamination in that case came from the air, right? These are both grown at the same temperature, so you got similar growth patterns. So different organisms will grow differently, and we'll look at that. There's actually an organism we use in this lab that even changes color based on what temperature you grow it at. You grow it at 37 degrees, it's kind of a whitish color. When you grow it at 25 at a cooler temperature, it produces a red pigment, so it looks pink and then red depending on how much red pigment have been produced. So organisms can be significantly affected by temperature. And we know this. We use temperature against them a lot of times, don't we, when it comes to microorganisms. We're going to decrease temperature, right? And at their minimum temperature, they'll still grow, but their rate is going to be significantly reduced or even stopped, like in the case of freezing. They're going to grow at their optimum, their very best, right, as fast as they possibly can at that optimum growth temperature. And for us and for microorganisms that are pathogenic to us, that's 37 degrees Celsius, right? And that's if you give them a whole bunch of food and you give them everything else they need, they're going to grow like crazy. But then when you start getting much warmer, right, you run into problems. You get a pretty sharp decrease till you reach maximum. In this case, this can lead to death, right? When you increase temperature to a certain degree, even for ourselves, you are going to denature proteins that are needed for the metabolic processes of these cells. You're going to even damage DNA and membranes. You're going to kill cells. That's why heat is so effective for us in protecting us from disease. And this is why for certain foods they want you to go to a specific temperature, right? Because they know what type of microbes that are present commonly contaminate those food sources and what level of temperature would kill most of them, right? And so that's why you have those different degrees of temperature. That's why we cook our food. Other than most of us like the texture, right, changes that happen when you cook as well. I don't know about you guys, but raw eggs, I just can't do it. Texture is just not there for me. So there is a range of preferred, and we're right in the middle. We're the mesophiles, so are the pathogenic organisms for us. There are others that can grow at much higher temperatures. They have adapted to that. They wouldn't cause problems for us because in that case we're too cool. So your thermophiles are growing in your compost heaps and your hot tubs and stuff. They're not necessarily going to make you sick, unless they produce toxins that you could be exposed to, and I don't know of any that do. Your hypothermophiles, again, the hot springs in Yellowstone National Park, the hydrothermal vents, and the ocean floor. They're actually microorganisms living at those very high extreme temperatures. Then the cooler ones, some of these we have to worry about. Some are psychotrophs, can grow at even refrigerator temperatures, right? Like listeria. Some of them can produce toxins that can make you really sick, right? So they'll grow in the refrigerator, they produce those toxins, you ingest that food, and it can make you really sick. But you yourself are too warm, they can't grow. Although listeria, right, it can grow at refrigerator temperatures, it can also grow at the temperature of your body. So that's a scary one. Psychophiles are things in glacial lakes, right? So if you were to, say, ingest a fish from a glacial lake, right, it might contain microorganisms. But again, you're going to be too warm for that to actually grow in you. It's only if that organism produced toxins that you would then maybe ingest would it make you sick. Psycho actually means cold. And as far as I'm concerned, cold is crazy. I grew up in Massachusetts for 20 years. I was cryopreserved. That's why I look so young. I was frozen. My dad was the Yankee. My mom was the Southern Belle. And we have the Vietnam War to thank for me. That's how you end up with a Yankee and a Southern Belle. It's called war. In the military. In their case, the Air Force. So, how do we make things sterile, right? What do we have to do to make things sterile? We have an autoclave machine, which is high pressure steam. And this is effective at even destroying endospores that some bacteria can produce. So this is an example of one. We have them over, we share our sink with the biology lab, 107E if you were in there. Yep, those weird looking machines in the back of the room, those are our autoclave machines. So if you're curious as to what they look like, walk by 107, you'll see them in the back of the room. Miss Paula tries to run that when there isn't classes, right? Because it's warm, it's smelly, it's yucky, right? As she said, she loves killing all our bacteria. She was out for a little while this summer, so I mean, it took her like a week to destroy all the microorganism cultures that we had generated over the summer while she was gone. Poor thing. But the good thing is that we had a break before you guys came back, so she was able to kill it all before students started classes. So we do that. Any of the stuff that we contaminate or needs to be disposed of properly goes on that cart by the door, and Miss Paula comes around and takes it and autoclaves it and disposes of it properly. So that's our discard cart there by the door. There have been some people sometimes that will use our lab, and I walk in and they put drinks on that cart. I'm like, that is the dirtiest place in this entire lab. Although this is probably the cleanest lab on campus. Like sometimes we'll have meetings in here, I'll whip out my lunch and start eating, right? This room gets disinfected more regularly than any other classroom on this campus, right? I don't even know when and if they ever clean those desks, right? So, again, I'm not working with the culture while I'm eating, right? We're just having a meeting. And I know that it's clean. Cleaner than most places. Okay, so why don't we do this for our desktops? Why don't we pick these guys up and go bring them and autoclave them? One, they're not going to fit, right? Definitely not going to fit. Do they have large numbers of microorganisms? Not large enough that we would need to autoclave. Okay, but they do have quite a bit of organisms, which is why we disinfect with Lysol, and why we don't set things like the lids and the caps to our Petri dishes onto these, because we would pick up contamination. Make sense? All right, so as we've already shown, that this is going to be a source of contamination, right? So we need to limit our contact with the desktop. So our light microscopes, right? You guys should hopefully be familiar with most of the parts of the microscope. We will go over and take out our microscopes and actually use them next week. They're going to allow us to see what we want to see in this lab. Right, so we're going to use them quite a bit. The first two lenses you look through are referred to as the oculars. These magnify your image only once, but the image is split so that you have a binocular instead of a monocular scope like I grew up with. You are going to have your own scope, so you can adjust these for the width of your eyes, right? And if you're having trouble with that, I can help you by lining up the light with your pupil of your eyes so that you can see just one round image, right? You should not see binocular like you do in cartoons, right? And you want to get in the habit of doing that because you don't want to be squinting. I won't adjust your scope. I will look through or squint when I come over because I'm only looking for a second. You're going to be observing things much longer, right, and doing drawings or taking pictures or whatever. Your virtual image that you're looking at when you look through those oculars has been produced by the different magnification of your different lenses, right? And so the real image is produced by the objectives that we have several to choose from on a revolving nose piece. Please use the revolving nose piece. Don't grab the objective lenses themselves. They're screwed into that nose piece. If you keep grabbing them, they eventually come unscrewed, fall out, and break. They produce the real image, which is then magnified to produce that virtual image that appears like inside the microscope, right, kind of hanging in space. Your condenser is another lens. It's found in your stage. It condenses the light from your light source, in this case a lamp, a bulb, right, onto your specimen. Why don't we, when we're calculating magnification, consider the fact that you have two oculars? In fact, you have two, right, but it's only magnifying once. Right? So the image reaches your eye has only been magnified by two lenses, the objective lens and the ocular lens, but then it's split, right, so that you can view it comfortably with both eyes. Each one of your lenses that magnify your ocular or your objectives have stamped on them somewhere their magnification. X stands for times magnified. That's the units for magnification. When you want to figure out the total magnification, since we're using two in series, you times them. The ocular is set, right? And I have to say, they just made up these weird numbers. I don't think any of these exist in your lab manual. I've seen 20x objectives, oculars. I've never seen 15. And these are really weird numbers, like 45 and 97. I think they're just trying to demonstrate the concept of the math, right? You just multiply the two. So our microscopes, on the other hand, are represented here. So we have what we call the scanning lens. That's a 4x objective. It is denoted by having a red ring on it. After next week, it's pretty much useless in microbiology lab because the magnification is only a total magnification of 40. That's nothing when you're talking about microorganisms. It's a waste of your time. But it is very beneficial in that it creates a big gap between your stage and your slide. And it's kind of a good point to take your slide off and you don't have to worry about whacking into your objectives. Your yellow, what we call low power 10x objective times the 10x ocular, total magnification of 100, is where you will typically start, especially when we start working with bacteria. This is going to be your go-to. Just skip over scanning, go right to low. High dry is also a useless objective lens here in the microbiology lab. It's not enough magnification still. It's only 400. Dry means you do not apply oil to this. Most of the ones in this lab are blurry because students don't follow directions and properly clean their microscopes, and they leave oil on this lens, which should never had oil on it to begin with if you follow instructions, and it creeps into the objective, gets in behind it, and creates the blurriness that cannot be removed. Right? So you'd have to throw away that objective lens and get a new one. We don't really use that one, so the fact that it's blurry, who cares? Just move on to the next. The oil. You're going to add a drop of oil to your slide. Then you're going to move the oil objective lens into place. It's going to almost touch your slide. It's not going to touch it or break it if you're truly in focus at low. You can go right to low to oil, and it should not hit if you're really in focus. A lot of students sometimes are like, Miss Erica, I can't see anything. I said, yep, I know. Even from my desk, I can see a big gap between your oil objective lens, right, and your slide, which means you're not in focus, right? When you're in focus, that oil is going to fill that gap. It's going to be almost touching. And that's going to help focus the light at that higher magnification that you need to be able to see your image. Even though they're going to seem teeny tiny. 1,000 times magnified. Alright, so our go-tos are our yellow and oil immersion. Low power, and then oil immersion, high power. Highest power. Resolution is important, how well you can see your image. Trevor, do you need your glasses to see the board? No. Okay. No other glass wearers, huh? I don't have anybody to pick on today. Okay. Oh, do you need it to see the board? Do you? Okay. All right. Take them off for me. How many fingers am I holding up? Okay. Put your glasses back on. 3. Right. What was the problem? They were close together, right? If I had done this, you might have been able to guess 3, right? And again, her resolution is off because the lenses in her eyes can't adjust anymore or never could. And so she wears corrective lenses to correct for that, right? Our microscopes are fixed just like your lenses in your glasses have a prescription set for your eyes for that adjustment. So do our lenses in our microscope. They have a set ability of focusing light, right? And that number is right next to, on an objective lens, its magnification. So you see this one says 10x/0.4. That's its numerical aperture. You can think of it as its prescription. How good it is at focusing light and doing its job. Your condenser also focuses light, right? So it has a numerical aperture. It doesn't have a magnification, right? It doesn't do magnification. It just focuses light. And so you can see this one, it has it stamped on here. 1.25 is the numerical aperture, the ability of that lens to focus light. So resolution is that clarity, right? That issue we just had a moment ago where she could not clearly see that I was holding up 3 fingers because they were too close together. And they blurred into what she thought was maybe 2, right? She guesstimated on what you know, right? Your brain really factors into a lot of what you see too. And can kind of throw you off sometimes too, right? Resolving power of that limit of resolution can actually be calculated mathematically. So it's a simple formula that you do unfortunately have to memorize and be able to calculate. And will give us an actual number and help us to determine if you would be able to see things as separate or blur together. So it is the wavelength of light over the numerical aperture of your condenser, right, because it focuses light onto your specimen, and whichever objective lens you're using to magnify. So NA stands for numerical aperture, D stands for resolution or resolving or limit of resolution. So with resolution, we actually want a small number, right? We want to be able to get them really close together and be able to see. And so the smallest fraction is when the top, right, is the smallest, our wavelength. So we use blue light filters because they have the smallest wavelength of light. And so therefore going to give us the smallest resolution. Remember, for your microscopes themselves, the numerical aperture for each of those lenses is fixed. It cannot be changed. It is a glass lens, it's stuck where it is. You can't change it like we can the lens in our eyes. Can't change the shape. So, of course, as we increase in magnification, so does the resolution. Otherwise, we wouldn't be able to see. So small is good. So don't be gluttonous, right? So 1/6 is what we want when we talk about resolution. We only want that one slice of pie, right? Because the top of the fraction is 1. We don't want the other leftover portion, which is 5/6 of the pie. Right? That's a really large number, much larger than 1/6, and it would give us a very poor resolution. So wavelength of light, that top of that fraction is really important. But the problem with light microscopes, we have to stay within this range, right? Called visible light. If we get outside the visible light, can we see it? No, right? You could use ultraviolet light, but we can't see that. That would give you really great resolution, right? But we can't see it. We have to stay in our spectrum, right? And blue is the best, right, within the spectrum for increasing resolution. So all our microscopes have blue filters on them. So we're using that wavelength of light to help us visualize. It won't make it look blue, right? But we're using mostly blue light. So for a given microscope, if they give you numerical apertures and you know what wavelength of light, basically what filter you're using, you can calculate resolution as a number, a specific distance in nanometers. So following our formula and the information given, right, we'd have 520 over 1.25 plus 0.25, which gives us 347 nanometers. That's the minimum distance, right? I'd have to hold two objects apart and you would see them as being separate. So having said that, that's our magic number, 347, right? Anything less than that, it's going to blur, right? They're going to be too close. So in this case, 330, are we going to see them? No, they're too close, right? They're less than 347. They're going to blur together. All right, so now let's change objective lenses. All right, so do we have to change our does our numerical aperture change? Yep, the lens change. Does the resolution change? Yes. All right, the numerical aperture changed. So now we have, instead of 0.25, we have 0.85. The condenser doesn't change, right? That one always stays the same. So now we've gone up in magnification, we have better resolution, we can put them 248 nanometers apart and we'll see them as being separate. So if two things are in fact 250, will we see them as being separate? Yes, right? It's greater than 248. That's the key. That's your limit, right? Anything less than that you won't see. Anything at that or greater you will see as two separate images based on that resolving power under those conditions. Now for our microscopes, we're going to pretty much live under oil. Right? You only have to clean off the oil when you're done, not in between each slide when you're done for the day. You wipe it off your lens so it doesn't creep in like it has for the high dry. It's only when I check to make sure you've done. So for ours, our filter is at about 500 nanometers. The condenser and the numerical aperture of the oil immersion lens is the same, 1.25. So that gives us a resolution of about 200 nanometers, or 0.2 micrometers, which is pretty small. It's definitely past the limit of our eyes. Our eyes, this is a ruler. Each one of these little lines is a millimeter. Apparently we can see down to 1/10 of a millimeter. I don't know about that. But in comparison, right, at 100 times objective lens, right, at 1,000 times magnified, with the resolution of 0.2 micrometers, it's 500 times what we can see with our own eyes. Pretty impressive. So this is the types of things that you'll see. Good old bacteria, right? These are actually rod-shaped in chains. Here's some yeasts that are even budding. Here's a red blood cell and a cluster of cocci-shaped bacteria. This is a super cool website that one of my students turned me on to a while ago. It's interactive. You can, and I'll actually probably post the individual link to this for you guys. It does a comparison of size. It even has, right, the different measurements down from meter, centimeter, millimeter, micrometer, nanometer, angstrom. And they're embedded within this. So you'll notice a grain of rice as compared, a grain of salt as compared to a cute little amoeba and paramecium, right? In the human egg, the largest cell in a woman's body. All sperm are mobile DNA. All the food and everything is in the egg. That's why it's so big. It's a single cell. Even an ostrich egg is a single cell. Half the DNA at that. Skin cell, right? Red blood cell. Baker's yeast, right? This is eukaryotic, pretty big, right? We got to zoom in even more before we get to bacteria, right? Like E. coli, That poor guy is smaller than even one of our mitochondria, one of those organelles in our cells, right? And then that's past bacteria or past the limit of a light microscope. You'd have to use an electron microscope to see like these viruses. And to be able to go down to the atomic level, which we have the ability to do now to see molecules like antibodies and hemoglobin, all the way down to a single carbon atom, which is the basis of all life on Earth. That's why we can do carbon dating. Most of your molecules have carbon as the backbone molecule, right? We have microscopes that can go to this level now, atomic force microscopes. Pretty impressive stuff. So we use bright field microscopy. This is where the background is going to be bright. The organisms are either going to have color to them or we're going to stain them. That's going to create the contrast for us to see them. This is a cute little euglena. He has his own colorization. This is dark field microscopy using different filters or directing the light in a different angle. So therefore the background is dark. That's why it's called dark field. The field is dark. And the organism itself will typically be illuminated, more so, as you can see, shiny here as opposed to here. Phase contrast, something I would like for us to have in our lab, utilizes, again, special filters and helps with light and allows us to see subtle differences in how light travels through different substances like the cytoplasm. And so you can see this same euglena, quite a bit more detail here for the phase contrast. Right? You can see all these little guys here that you didn't even quite notice in these other pictures taken with the different type of light microscopes. And then, of course, my favorite, fluorescent microscopy, which has become very, very popular as of late. Some organisms are naturally fluorescent. When you shine UV light on them, they'll fluoresce. We're actually going to make green glowing E. coli, fluorescent E. coli in this lab at the end of the semester. We don't have special microscopes though to look at them, but we'll be able to look at them growing on a petri dish, large number, and shine UV light on them, and they'll be green. These could be naturally fluorescent, or we can use specially tagged dyes that have fluorescence to them, even to the level where they're linked to, say, an antibody and will bind to a specific component on a cell. So I mean, we've gotten really advanced in this technology. These type of microscopes have to have special filters on them, of course, because they utilize UV light, and that would damage your eyes. In my office, I have a really cool poster from one of the talks that I went to, and has a bunch of different pictures of using different stains, and they arranged it in a rainbow. Right? So science is, we use colors a lot, especially in microbiology. Different colored tags, and it can be as gorgeous as some of the most beautiful art that you see out there. Getting to see pictures of these things stained. Pseudomonas is another organism we use that produces a green pigment that is actually fluorescent. So when you shine UV light on it, it fluoresces. It fluoresces blue. It's really pretty. So that's it for today. And